Dear friends, this video is the continuation of my biochemistry lecture series. In my previous video, I have explained the amino acids, which are the building blocks of proteins, and today I will explain the plasma proteins in detail. Plasma protein is a very important topic in the clinical biochemistry because they are used for the diagnosis of different diseases. We can say that these are the biomarkers for different diseases. Their imbalance in the blood can diagnose the multiple disease conditions. The learning objectives of today's video is to understand the properties, functions of different plasma proteins, their characteristics, and their clinical relevance. The other most important objective is to understand the separating methods of the plasma protein because separation methods are very important. Without standard separation protocols, we cannot quantify the proper quantity of proteins in the plasma, and we can misdiagnose the diseases. We can only analyze the plasma proteins after the separation. Therefore, separation methods are very important. Plasma is a pale yellowish liquid that we get after the separation of red blood corpuscles, white blood corpuscles in the blood. Plasma is actually a 70% of the blood, and among this 70%, 7 to 9 gram is plasma protein. Human blood contain both type of proteins, conjugated and simple. Simple proteins are those which are only purely made up of protein molecule, and conjugated proteins are made up of two different types of molecules. One is protein and the other is any carbohydrate, lipid, or any other molecule. The most commonly found conjugated proteins in the blood are glycoproteins and lipoproteins. In lipoproteins, any kind of lipid is attached with the protein molecule, and in glycoprotein, any type of carbohydrate molecule is attached with the protein molecule. Normally, we get 80 different types of proteins in the human blood plasma, and most of these proteins are transport proteins because blood performs the function of transporting the minerals and nutrients, hormones, enzymes. Most of the enzymes and hormones are proteins, so we normally get transport proteins in the plasma. There are three major plasma proteins. These are albumin, globulin, and fibrinogen. Albumin is most abundant, is a major plasma protein which is 50% of the total 70% of the blood. However, globulin is 29%, but globulin also has four types: alpha 1, alpha 2, beta, and gamma globulin. The third most important is fibrinogen, which is 11% of total 70% of the blood, and this protein is one of the clotting factor of the blood. Now coming to the clinical relevance or the significance of the plasma proteins, the biggest clinical relevance is these are the biomarkers. Markers are those chemicals or things, products which are used to diagnose any disease. When the markers, any kind of marker is disbalanced in the blood, we can suggest or we can diagnose the disease by analyzing those markers. So plasma proteins are one of the markers of the multiple diseases like multiple myeloma, in which beta and gamma globulin is increased, and M proteins, which are called monoclonal proteins, which are the abnormal type of immunoglobulins like antibodies. These are mostly increased in the disease like multiple myeloma. Multiple myeloma is a cancer, type of a blood cancer. Likewise, rheumatoid arthritis. Rheumatoid arthritis is an autoimmune disease in which alpha globulin is increased. And this chart shows the multiple bands